welcome to this workshop on flow cytometry. It's amazing to see all people here are interested in flow cytometry. And flow cytometry is a very powerful tool to work with a cell. And you can use it in different ways. You can work with the cell from receptor, cellular signaling pathways. Some they can do sorting. You can sort the different types of cells. You can study their function, their proliferation. You can study the DNA. You can study the different phases of cell division. It's a very powerful. There are a lot of technology. I learned to work with fax. We used to call it fax in 1988 when I was a student. And at that time, I saw it as a complicated, but since the development of technology, it has become really very powerful and useful tool. And also in diagnostics beside research. This morning we have the great pleasure and honor to have Professor Volker. He is the head of the flow cytometry core facility in Heidelberg University in Germany. And he is one of the world experts in flow cytometry. And he has been also in the region for some time. He was a visiting professor at King Abdulaziz Medical City for Science and Technology in Riyadh, and also in biotechnology program at the Graduate College at AGU. And thank you so much for being with us, complete 1 week. And he's also supervising Dr. Huda on her PhD thesis on studying some molecular aspects of infertility. Today, he will give you some theoretical background on the uses of flow cytometry, and Mrs. Sala will show you some of the practical issues for the machine itself in the lab to see in live what he will tell you. I wish you a good working day and enjoyable time.
There will be handout. This is our surprise. Handout.
Okay. Yeah, I saw the handout. What was preparing. Okay. Good luck and have a great day and a great weekend.
Yes.
See you.
Thank you, Professor Weiss.
Thank you.
Good morning. It's a great pleasure to see a lot of people which are interested in flow cytometry. It's not usual. And I will give you a first theoretical applications in flow cytometry. And I will start with a history. The flow cytometry was developed from the normal microscopy. And 1947, it was built the first prototype of a flow cytometer. Kaminsky and Fulweiler built the first flow cytometer and also in cell sorter in 1965. Wolfgang Goethe developed the first flow cytometer in Germany. It was 1969. And I myself started 1970 with the first Patek flow cytometer in Europe, in my lab in Heidelberg. 1962, Herzenberg constructed a flow cytometer, which is called the fax. And the fax is a synonym for fluorescence activated cell sorter. The flow cytometry integrates electronics, fluidics, computer, optics, software, and laser technology in a single platform. The flow cytometry uses the principle of light scattering, light excitation, and emission of fluorochrome molecules to generate specific multi-parameter data from particles and cells in the size range of 0.5 micrometer up to 40 diameter. That means you have the chance from large cells going down to yeast. Also yeast you can detect it by flow cytometry. Cells are hydrodynamically focused in a sheet of PPS before intercepting an optimal focused light source. And the light source are lasers or HBO 100 lamps. Cells are presented to a laser using principles of the hydrodynamic focusation. You see here the chamber, the meshing chamber, the flow chamber. On outside there is a sheet fluid, which is PPS, and in the middle is running your cell population. And at this point here, one cell after one cell going directly to the laser beams, which are detected each cell by each cell. This is the principle of hydrodynamic focusing. As your cells or particles of interest intersect the light source, the scatter light and fluorochromes are excited to a higher energy state. As the molecules relax to a lower state, energy is released as a photon of light with specific spectral properties unique to different fluorochromes. Scattered and emitted light from cells and particles are converted to electrical pulses by optical detectors. Collimated light quanta is picked up by confocal lenses focused at the intersection point of cells and the light source. And the light is sent to different detectors by using optical filters. For example, a 525 nanometer bandpass filter placed in a light pass prior to the detector and will only green light detected. The most common type of detectors used in flow cytometry is a photomultiplier tube. And this photomultiplier tube converts light into electrical pulses. And these electrical pulses originated from light detected by the PMTs, from photomultiplier tubes, are then processed by a series of linear or logarithmic amplifiers. So logarithmic amplification is most often used to measure cells, the fluorescence in cells. And this type of amplification expands the scale of wave signals and compresses the scale for strong or specific fluorescence signals. After the different signals or pulses are amplified, they are processed by an analog digital converter. which is in turn allows to event to be plotted on a graphical scale, one parameter, two parameter, or multi-parameter analysis. The optical filters found in the most flow cytometry can be several types and knowledge of the ones that are used or that we can use will aid in the design and the feasibility of multicolor experiments. And this is an optimal bench thematical. You see here the flow cells running by this way. The laser beam is come from side, and you see several detectors for forward scatter. Forward scatter means you can detect the size of a cell. The SS sensor, the side scatter, detects the granularity of a cell. And you see filters with different wavelengths. For example, the flow FL1 sensor is a bandpass filter, and you can detect by this photomultiplier, for example, FITC colored cells. Or the FL2, you can detect phycoerythrin colored cells. It's an orange color. And the FL3, you can detect propylium iodide. Or what else? APC, for example, colored. And this detects red cells. And you see by filters which are built inside, at first all colors will come through these filters and selected by the green. The red or orange is going through this filter, detected by the FL2, and the red colored is going through this filter system and detected by this photomultiplier. That means you have so-called long-pass filters, you have short-pass filters, it's band-pass filters, and decoid filters or mirrors. And this is a fluorescence. You have a cell, and antibodies recognize specific molecules on the surface of some cells, and the antibody will catch the outer surface of a cell, and you can detect these cells with the antibodies and the color of FITC, and you can analyze, when cells are analyzed by flow cytometry, the cells expressing the marker for which the antibody is specific will manifest the fluorescence, and cells who lack the markers will not manifest by fluorescence. For an example, a two-parameter histogram, you see on the x-axis, you see the forward light scatter, which is marked the size of a cell, and the y-axis is the side scatter, which are marked the granularity. And by this analysis, the size against the granularity, you can analyze, for example, living cells or dead cells, or on the way to the dead by apoptotic cells, and you can also detect bigger cells such as tumor cells. by coloring cells with two colors. For example, FITC and phycoerythrin, you can make a quadrant analysis, which is described here. You see on the left corner negative populations for both, for FITC and also for PE. Single positive populations are located here. Single positive cells which are marked with phycoerythrin is located here. And in this quadrant we see double positive cells. Cells which are marked by FITC as well by phycoerythrin. This is most analysis by surface antibodies for analyzing, for example, blood. The DNA of cells can be stained by a large number of dyes, such as propylidomide, ethylbromide, PEG stains, mitramycin, and the most common is Darby stain. The Darby stain has a benefit that are not intercalating in the DNA, in contrast to propylidomide. The Darby stain is only going to AT groups, in contrast to propylidomide or ethylbromide. And you see here a lot of fluorochromes which are common for analysis, cell cycle, surface antigens, and so on. And you see here the excitation. For example, dark here, it's a UV color, in contrast to propylidomide, as a green excitation and a red emittation. Only for to describe what's possible to mark cells. What is flow cytometry? Flow cytometry is a method for quantitative and qualitative measurement for nucleic acids, such as DNA and RNA, intracellular proteins, and also extracellular proteins. Flow cytometry is the only technology able to resolve molecular and functional markers on one cell at a time, impacting both for clinical diagnosis of human diseases and cell biology research. There's a lot of art in the science of cytometry. Since we have to keep working hard to adapt the tools to changing biological demands. Methodology of flow cytometry have uniquely impacted process in immunology, cell biology, cancer research, and clinical medicine. Single cell operation, marker and measuring is according to the standard of standard SOPs after GMP and GLP. GMP is good manufacturing practice and GLP is good laboratory practice. And this terminology is according to the Convention of Nomenclature for DNA Cytometry and the Committee of Nomenclature Society Analytical Cytology was founded 1984. And guidelines for implementation of clinical DNA analysis was founded in 1993. The application of flow cytometry is the immunofluorescence, cell cycle kinetics, cell kinetics, genetics, molecular biology for biotechnology, microbiology, biological oceanography, parasitology, and bioterrorism. Cellular parameters measured by flow cytometry, its intrinsic, no reagents or probes required structures such as cell size, we have heard the former light scatter, cytoplasmic granularity, size scatter, and photosynthetic pigments. The extrinsic, its unique reagents for structural, so DNA content, DNA base ratios, and the RNA content. And functional, surface and intracellular receptors for DNA synthesis, DNA degradation, for example, apoptosis, cytoplasmic calcium flux, and gene expression. And what can we measure? Cell cycle analysis are the most important. Immunity analysis is involved in this. Immunophenotyping for blood synthesis. Chromosome analysis, cell function measurements, and apoptosis. And what kind of material we can measure? So we can measure solid tumors, such as biopsies, cell cultures, for example, suspension cultures or adherent cultures, natural suspension blood cells, for example, blood. We can use native biopsies, cryoconserved, stored at -80 degrees, ethanol fixed cells, and for retrospective analysis, we can use histological embedded materials. But this method is to separate from histological embedded materials is very complicated and takes a long time to solve single cells from embedded tissue. But it's possible. But it takes a very long time. The measurement fluorescence is proportional electron impulses able to an exact and very fast acquisition of deltas. At the end of a measurement displayed in a histogram, or a dot plot, or a density plot, or a contour plot, analysis can be done. The software for acquisition and analysis, flow cytometer for the particle system. The FlowMax software is installed in particle machines. Also, the multicycle and the WinMDI software. For BD machines, the software is Simquest, as you have here in your lab. The key factors of DNA probes is that they are stoichiometrical. Thus, numbers of molecules of probe bound is equivalent to to the number of molecules of the DNA. One of the earliest applications in flow cytometry was a measurement of DNA contents in cells. This was the beginning of the flow cytometry. The first technique available to characterize the non-mitotic phases of the cell cycle. This analysis is based on the ability of certain dyes to stain cellular DNA in a stoichiometric manner, which is directly proportional to the amount of DNA in a cell. In this picture you see the cell cycle. It's starting with the first growth phase, so-called G1 phase. DNA replication is in the S phase. Growth in preparation for mitosis is in the G2 phase. The mitotic phase is called in the M phase. The mitotic phases, O phase, metaphase, anaphase, and telophase, it's not possible to measure by each step. It's in general the M phase. The G1 and 0 phase is where the cell spends the most the most time of their life. And all cells of the same species contain the same amount of DNA in the G1 phase. And this is called 2n amount of DNA because there are 2 copies of each chromosome in the nucleus of the cell. When a cell gets ready to divide, it must first duplicate its DNA. So when it's divided to each daughter cell has the same amount of DNA as the original cell. This duplication of the DNA is called the S phase for synthesis. One of the cells has finished synthesizing its DNA. It has twice as much as the original cell, so contained 4n of DNA. This stage of the cell cycle is called the G2M phase. Next, the cells goes through the mitosis, which split the 4n amount of DNA back into 2n, and cells returned to the G1 phase. Some are in G1 phase, while simultaneously some are in S phase and G2 phase. For this reason, we get the distribution from the flow cycle that contains all phases in a cell cycle. This type of data requires the cell cycle analysis software to analyze because of the overlap of the S phase and the G2 and the G1 phase populations. In addition, if there is debris of or the overlap of a second cell cycle, which is called aneuploid in the DNA, a simple analysis with markers on the histogram is highly inconsequential. DNA content histogram requires mathematical analysis in order to extract the underlying G1, S, and G2 phase distributions. And this method for analysis have been developed and refined over the past 2 decades. The most flexible, accurate method of cell cycle analysis is based on building a mathematical model of DNA content distribution and say fitting this model to the data using curve fitting method. The most established model was proposed by G and Scheck in 1974. Almost all cells or nuclear suspension analyzed by